The water here is close to freezing, not the theatrical cold of an ice storm or a winter river, but the deep, still cold of a place where the sun has never reached. A cold that has not changed in thousands of years, that holds everything in it motionless and exact. 2 degrees above freezing, sometimes 1. The pressure at this depth would collapse a human lung in an instant, but the water does not feel hostile. It feels ancient. It feels like a room that has been sealed for a very long time, and the air inside it, the water inside it, has settled into something close to permanent. Somewhere on the seafloor, 400 feet below the surface of the Gulf of Mexico, a small opening in the sediment releases a thread of fluid into the cold. There is no eruption, no plume of superheated water, no thermal shimmer, no roar, no violence of any kind. The fluid simply rises, barely warmer than the water around it, carrying with it dissolved gas from the rock below. A single bubble forms at the opening, then another and another. They catch the faint light of a research instrument descending from above, a light that has never been here before in the history of this place. And each bubble holds that light for a moment as it lifts away from the sediment and begins its long, slow ascent toward a surface it will never reach. The gas dissolves back into the water long before that. The ocean absorbs it. The seafloor exhales, and no one above notices. This is a cold seep. The name is quiet and unglamorous, and that is fitting because the thing itself is quiet and unglamorous. No heat, no fire, no dramatic geological event, just fluid, rising, gas, migrating upward through sediment that has been accumulating for millions of years. Methane, mostly, the same gas that forms beneath swamps and rice paddies, beneath landfills and the slow-rotting floors of shallow lakes. But here it comes from much deeper, pressed up through the compressed layers of the continental margin by forces far older and larger than any single landscape. It seeps rather than erupts. It rises rather than blasts. And in doing so, it creates something that takes a long time to understand, because the life it supports is built on a chemistry invisible to the eye and a timescale that dwarfs the observer entirely. There are other places on the ocean floor where hot fluid rises, hydrothermal vents, cracks in the crust where the planet's interior heat finds a path upward. The water there can reach 400 degrees Celsius and still not boil, held liquid only by the crushing pressure of the deep. It comes out black with dissolved minerals, building chimneys of sulfide and oxide rock, dozens of feet high. It comes fast, turbulent, energetic, a geological event unfolding in real time. The vent communities it supports are spectacular in a way that registers as spectacle, even to a human observer seeing them on a screen. They feel dramatic. They feel active. Cold seeps feel none of those things. The temperature at the fluid exit is within a degree or two of the surrounding water, so close to ambient that no instrument trained on heat would flag it as remarkable. The fluid does not roar out. It seeps. Where a hydrothermal vent announces itself in a plume of black or white mineral-rich water, visible from hundreds of feet away, a cold seep announces itself only in what grows around it, the pale mats, the unusual shells, the carbonate rock in the life that has accumulated over years and decades in response to the quiet chemical gift the sediment provides. The contrast is the difference between a geyser and a spring. Both bring water from underground. One is a spectacle, and one is a place where something quietly, persistently lives. Understanding why the fluid rises at all begins in the geometry of the ocean floor itself. The seafloor is not a single flat surface. It is a collection of geological structures, each with its own history of formation and deformation, and along the edges of continents, the continental margins, the seafloor is old and layered and compressed with organic matter that accumulated over vast spans of time. Organic material settled through the water column, reached the floor, and was buried. More organic material settled on top of it, and more still, layer after layer after layer, the weight of the above pressing on the below, the oxygen draining away as burial deepened, and in the oxygen-depleted darkness of that burial, something quiet began to happen to all that organic matter. Microbes decomposed it, carbon compounds transformed. Over enough time and enough pressure, the organic material yielded gas, methane primarily. It formed in the pore spaces between sediment grains, accumulated slowly, and began to look for a way out. Along the convergent margins of the ocean, the places where one tectonic plate slides beneath another, the geometry of the situation accelerates this process in a specific way. A slab of oceanic crust moves steadily toward a continental margin. It descends. As it descends, it drags the sediment that has accumulated on its surface down into the zone where the two plates press against each other. The sediment is wet, saturated with pore water, the fluid trapped between sediment grains, and as the pressure of convergence increases, that pore water has nowhere to go but up. The sediment piles up at the margin in a feature geologists call an accretionary prism, a wedge of compressed material building outward from the continent, like snow piled against a shovel blade. Within the prism, thrust faults form, fractures along which the compressed material has slipped and shifted, and these faults become pathways. The methane-rich pore water follows them upward. It finds the seafloor. It seeps. The margins of the Cascadia subduction zone off the coast of the Pacific Northwest hold some of the best studied of these convergent seep systems. So does the Japan Trench and the Makran margin off Pakistan and Iran. These are places where the planet's tectonic machinery, the long, slow, irresistible movement of crustal plates over a partially molten mantle, happens to intersect with the right combination of sediment thickness and organic richness to produce fluid that migrates, finds pathways, and exits at the surface into water that has been waiting for it for longer than there have been humans to wonder at it. Convergent margins are not the only places this happens. On what geologists call passive margins, the quieter edges of continents, where no active subduction is occurring, where the ocean floor simply meets the continent without the drama of two plates colliding, cold seeps form through entirely different mechanisms. Passive margins are places of slow, steady geological change. The continental shelf here is broad and shallow, and the sediment that has accumulated on it over millions of years is thick, sometimes kilometers thick, and its own weight presses it downward, compacting it, squeezing the fluid from its pores, driving that fluid upward toward lower pressures. Gravitational sediment compaction is the name for this process, and it is exactly as slow and as patient as that name implies. On some passive margins, there is another mechanism at work: salt. Buried beneath hundreds of meters of sediment are deposits of salt left from ancient evaporated seas, and salt, under pressure, behaves unlike almost any other rock. It flows. Over millions of years, it deforms and rises through the sediment above it, pushing upward in columns and domes called diapirs, and the movement of these salt bodies distorts and fractures the sediment around them, creating pathways through which methane migrates. The Gulf of Mexico is the great example here. Beneath its floor, ancient salt deposits left from a closed sea are still moving, still deforming, still creating the fractures and conduits through which methane has been rising toward the water above since long before the continent took its current shape. The seeps scattered across the Gulf's continental slope are in part the visible surface expression of that ongoing salt tectonics, quiet outlets for a vast, buried geological process. Mud volcanism is a third mechanism. In places where sediment accumulates rapidly and gas production within it is high, the sediment can become oversaturated with fluid and gas, and under the pressure of its own accumulation, it may mobilize and rise through the layers above it, eventually erupting at the sea floor in low, broad structures called mud volcanoes. These are not like the incandescent volcanoes of subduction zones. There is no molten rock, no ash, no lava. They are mounds of cold, saturated methane-rich sediment that has forced its way upward and pooled at the surface, building a gentle cone a few meters high over timescales of years to centuries. At their apices, fluid and gas release steadily. At their flanks, the sediment that has flowed outward creates a gentle slope of disturbed material that grades back into the undisturbed seafloor around it. They look, in the footage from a descending ROV, like low hills in an otherwise flat plain, modest and uneventful, doing what they have been doing for thousands of years, entirely without observation, entirely without consequence, beyond the slow chemistry they sustain. All of these mechanisms, the thrust faults of convergent margins, the gravitational compaction of passive margins, the salt tectonics of places like the Gulf of Mexico, the mud volcanoes that form wherever sediment is thick and gas production high, produce the same surface expression. Methane-rich fluid rises to the seafloor, and at the seafloor, something waits for it. The first thing visible at an active seep, if the water is clear and the instrument light is right, is often the bubble stream, a thin thread of gas bubbles rising from a small hole in the sediment, each bubble a few millimeters across, coated almost immediately in a thin film of dissolved material from the seawater around it, catching light and releasing it as it ascends. The bubbles do not rise straight up. They spiral slightly, they oscillate. The current, even at depth, is not completely still, and the bubbles follow it, drifting slightly in whatever direction the water is moving. They form a column that is not quite a column, more of a diffuse upward drift, visible against the dark background of the water if the light hits them right, but easy to miss if it does not. At some seeps, the bubble streams are vigorous enough to be detected by ship-mounted acoustic instruments on the surface, which read them as anomalous backscatter in the water column, unexplained patches of returned sonar signal that led researchers in the earliest surveys to wonder what was rising from the floor below. The bubbles are mostly methane. Some contain other hydrocarbons, trace amounts of ethane, propane, small molecules that formed alongside methane in the deep sediment. They are buoyant relative to the surrounding water, and they rise, but they do not rise far as pure gas. Within tens of feet of the seafloor, in most seeps at depths greater than a few hundred meters, the cold temperature and the high pressure conspire to form something else, methane hydrate, an ice-like lattice of water molecules holding methane molecules inside it. A bubble rising from deep sediment may develop a hydrate skin, a thin crystalline coating that slows its dissolution and lets it travel a little higher before the methane escapes into solution and the bubble disappears entirely. At the very deepest seeps, the hydrates are stable enough to form on the seafloor itself, appearing as pale, almost luminous patches of ice-like material nestled in the sediment, slowly releasing dissolved methane into the water above them. They look like something that does not belong on the ocean floor, something from a shallower, colder world, but they belong here. The pressure and cold of the deep sea are exactly the conditions under which they form and persist. Surrounding the bubble streams, or at seeps where there are no visible bubbles, covering the sediment in thick, continuous patches, are the microbial mats. They are white, mostly. White or pale orange, depending on the sulfur chemistry of the sediment beneath them and the metabolic state of the bacteria that form them. Seen close, they have texture, a dense, matted, fibrous surface, almost like a carpet or like snow that has partially crusted over. Better to think they do not move, though the gentle current moves the water above them. They extend across the seafloor in irregular patches that follow no obvious boundary, covering some areas densely and leaving others bare, tracing the hidden geography of the fluid rising beneath them. Where the methane flux is highest, the mats are thickest. At the edges of the seep, where the flux diminishes, they thin to a patchy scattering and then disappear, and the sediment returns to its ordinary gray-brown color, and the community they support gives way to the sparse, wide-spaced fauna of the general deep-sea floor. The carbonate rock forms slowly. Over years and decades, the chemical changes that the seep fluid produces in the surrounding sediment lead to the precipitation of calcium carbonate in solid form. It is not the smooth limestone of a tropical reef, polished and uniform. It is rough, angular, irregular crusts and slabs and nodules of pale mineral, precipitating at the boundary where the rising fluid meets the chemistry of the overlying seawater, locking the record of the seep's activity into the sediment layer by layer. A single slab of authigenic carbonate, carbonate that formed here, in place through the chemistry of the seep rather than through the accumulation of shells, may be only a few inches thick, but may record decades of activity in its mineral structure. The seafloor around an active seep becomes, over time, harder and more complex. Where once there was soft sediment, there is now rock. The rock outlasts the fluid activity. It persists after the seep has quieted, a physical record of what was once here. And scattered across this rocky, mat-covered seafloor, attached to the carbonates and anchored in the sediment, and clustered in dense groups around the most active vent openings, are the animals. Mussels, some of them dark-shelled, packed together in beds that extend for yards in all directions, their shells crowded against each other, their position at the seep center marking it as the most productive ground. Tube worms, in other locations, long white tubes rising vertically from the sediment, topped with feathery red plumes extended into the water. Pale clams, their shells half buried, their bodies bridging the sediment surface and the water above. Snails moving slowly across the mat surface. Crabs, pale and deliberate, picking at what the mats and the mussel beds provide. The seep is not empty. It is not even sparse. It is by deep-sea standards extraordinarily, almost improbably dense with life, a concentration of biomass in an environment that elsewhere supports almost nothing. None of this is accidental. All of it traces back to the methane rising from below, to the chemistry it enables, to the life that has adapted over millions of years to use that chemistry as a foundation for everything else. The mats are there because the methane is there. The mussels are there because the mats are there. The crabs are there because the mussels are there. And the methane is there because of faults and salt and sediment compaction and the slow, relentless weight of geological time pressing down on organic matter until it transforms. The seep on the floor of the Gulf of Mexico, with its bubble streams and its pale mats, is not alone. There is another one a few miles away, and another beyond that, and hundreds more scattered across the Gulf's continental slope at depths ranging from a few hundred feet to several thousand. And the Gulf is not alone either. Cold seeps have been identified on every continental margin of every ocean basin on Earth. The Atlantic margin of North America, where the passive sediment sequence is thick with buried organic matter. The Norwegian margin, where gas hydrates form in the cold deep water and methane bubbles upward through sediment deposited since the last glaciation. The New Zealand margin, and the Chilean margin, and the eastern margin of Africa, and the coasts of Japan and Indonesia. The Black Sea floor, nearly landlocked, where methane-driven communities persist in water so deep that it has been isolated from ocean circulation for thousands of years. Even in the deep trenches, the Haddle zones that descend beyond 20,000 feet into the seafloor, cold seep communities have been found. Their organisms adapted not only to the chemistry of the methane, but to the extraordinary pressure of those depths. New sites continue to be mapped. Each improvement in acoustic survey technology, each generation of ship-mounted sonar capable of detecting methane bubble streams in the water column, each new ROV capable of descending to greater depths and capturing footage of the seafloor with higher resolution reveals seeps that had been invisible before. The deep continental margin off the eastern United States was once thought to hold few active seeps. Then improved sonar surveys revealed hundreds of methane plumes rising from the seafloor. Some of those plumes, when followed down to the sediment surface, led to active seep communities. Some dissolved before reaching any visible vent. The full picture of cold seep distribution on Earth is still being assembled, year by year, survey by survey, each new voyage adding a point to a map that already covers every ocean and yet continues to grow. What all of these sites share, the Gulf of Mexico and the Norwegian margin, the deep Japan Trench and the shallow Hudson Canyon, is the fundamental character of the cold seep: fluid, rising slowly, temperature barely distinguishable from the ambient cold, a chemistry built from the deep burial of organic matter over geological time, a surface expression of bubbles and mats and carbonate and life that persists, quietly and without witnesses, in the cold dark of the deep ocean floor. The planet has been doing this for a long time, not just since the last ice age, not just since the age of the dinosaurs, but for hundreds of millions of years, since the continents began accumulating sediment on their margins and the organic matter within those sediments began its transformation. The chemistry is old. The life that uses it is old. The carbonate rocks that cold seeps leave behind have been found in geological formations from the Cretaceous period, laid down 100 million years ago, and now exposed in road cuts and coastal cliffs where the ancient seafloor has been uplifted by tectonic forces. Those ancient carbonates hold the same chemical signatures as the rocks precipitating today on the floor of the Gulf of Mexico, the same methane, the same reactions, the same slow, quiet process operating through a span of time that makes any individual observation of it, any ROV descent, any sample collection, any camera frame, feel like a single blink in a gaze that has been open since before the first grass grew. The seafloor holds these systems distributed across its vast, mostly invisible surface. Quiet, cold, persistent. The methane that moves through them comes from burial and compression, from salt and fault, and the slow press of the Earth's own weight. It rises through the sediment in threads too small to follow individually, gathering in pore spaces, migrating along fracture paths that took thousands of years to open, finding the seafloor eventually and releasing itself into the water above in streams of bubbles that catch the light for a moment before dissolving into the cold column of the deep ocean. And around each point where they emerge, on every margin of every ocean, the same community assembles. The same mats, the same shells, the same carbonate, slowly hardening the sediment beneath them. The same life, meeting the same chemistry, on every seafloor the planet has produced. Some of these seeps have been active for 1,000 years, some for 10,000. Some, in geological terms, are practically new, opened by a recent fault movement, a freshly mobilized salt body, a sediment slide that cut a new pathway through otherwise impermeable rock. And some have been flowing so long that the carbonate they have built lies in deep stratified slabs beneath meters of later sediment, recording seep activity from centuries before any human civilization developed the tools to recognize what they were looking at. The ocean floor is full of these records. Most of them have never been sampled. Most of the seeps they record are no longer active. The fluid has moved on to a new pathway, or the source rock has been exhausted, or the fault has closed. But the carbonate remains. The chemistry remains, locked in mineral form, waiting in the dark for a drill, or a dredge, or a submersible that may or may not ever come. There is a particular quality to the stillness of the deep sea floor that is different from any other stillness. Not the stillness of a room late at night, where the heating system ticks and a distant car passes and the building settles under its own weight. Not the stillness of a forest in winter, where the cold holds everything in suspension, but the wind moves and the branches carry snow, and somewhere below the frost line, the ground is alive with things working. The stillness of the deep ocean floor is more complete than either of those. The water barely moves. The temperature has not changed in years. No light has ever reached here from the surface. Not in this place, not at this depth, not since the seafloor was laid down from the accumulation of falling particles, tiny shells, and mineral grains and organic fragments drifting down through a kilometer or more of water over thousands of years until they settled here and were buried. And into this stillness, the seep introduces a kind of motion, not dramatic motion, not the motion of a breaking wave or a flowing river or even a slowly turning current, just the faint upward drift of fluid, the rising of bubbles, the imperceptibly slow precipitation of carbonate around the vent opening. The seep disturbs the stillness, but only just. It is quieter than any surface process. It is slower than most geological ones. It operates on the time scale of the sediment itself, years and decades and centuries of slow, continuous release, and in doing so, it makes a place out of a featureless expanse where the fluid rises, life gathers, the mats form, the shells accumulate, the carbonate hardens, and the deep ocean floor, which would otherwise be nearly bare, a cold plain of gray sediment with a handful of organisms moving across it in search of the few particles of organic matter that rain down from above, becomes at the seep dense, layered, rich with activity that does not depend on the sun, does not depend on the surface, does not depend on anything but the slow, reliable upward migration of methane from the rock below. This has been happening without witness on every ocean floor the planet has produced, on the floor of ancient seas whose names we do not know, whose continents dissolved back into the mantle before the first vertebrate swam through them, on the floor of the Tethys Sea which closed as India moved north and pushed up the Himalayas, on the floor of the Proto-Pacific and the inland sea that once covered the center of North America. All of those seafloors held cold seeps, almost certainly held the same bubble streams and white mats and carbonate crusts and improbably dense communities of life built on the chemistry of buried organic matter. Most of that evidence is gone now. The ocean floor is recycled continuously by the same plate tectonics that drove the seeps in the first place, swallowed back into the mantle at subduction zones before anything on the surface has a chance to notice. What remains is only the small fraction of ancient seep carbonate that ended up on passive margins, incorporated into rock sequences that were eventually lifted above sea level by tectonic forces and exposed on continental land surfaces. Those outcrops are scattered across California and Japan and Norway and other places where old ocean sediment has been brought to the surface. They are visited by geologists who recognize them for what they are, records of cold seep ecosystems from deep time, and sample them and read in their chemistry the same methane oxidation signature that active seeps produce today. The same quiet chemistry, the same slow rising, the same gathering of life around a chemical gift from below. Across every ocean basin, on every continental margin, at every depth from the edge of the shelf to the deepest trenches that extend down into the seafloor, 7 miles below the surface of the sea. The seeps are there now, and they have been there for as long as there have been continental margins to hold them. They are not rare. They are not unusual. They are one of the deep ocean's most enduring and widespread features, present everywhere, visible to nothing, sustaining a world that has been building and rebuilding itself in cold, still darkness since long before the first creature emerged to breathe the open air. The bubble stream from that small opening in the Gulf of Mexico sediment has been rising since before anyone descended to see it. It will continue rising after the last recording instrument is brought back to the surface and the ship sails home. The methane from that opening does not know it has been observed. The carbonate forming slowly around the vent opening does not know it has been sampled. The mats and the mussels and the slow pale crabs do not know that the light of an ROV has passed across them once and will not return for years. They continue. The fluid rises. The seafloor exhales. The cold holds everything steady in its ancient, patient dark, and the deep ocean continues, as it always has, quietly alive. Deep inside the sediment, beneath a kilometer of cold ocean water, organic matter is being slowly unmade. It has been settling for a long time. Fragments of once living things, the broken shells of creatures that drifted near the surface, the soft tissues of animals that sank when they died, fine organic particles carried by slow deep currents, all of it has been accumulating in layers, one thin film over another, pressed and compressed by the weight of everything above. In the topmost layers there is still some oxygen, dissolved and diffusing in from the water above. But a few centimeters down the oxygen runs out, and what remains is a sealed, lightless, airless world, where an entirely different kind of chemistry takes over. This is where methane begins. In the oxygen-free layers, microorganisms that cannot tolerate oxygen carry out a slow dismantling of the organic compounds buried around them. They feed on what settles down, processing carbon compounds and releasing the simplest possible product. Molecule by molecule, in cold darkness, carbon is being converted into methane gas. The process is not fast. It takes time measured in decades, centuries, or longer, depending on the depth and temperature and the rate at which fresh organic material arrives. But it is continuous and vast, playing out simultaneously across the sediment floors of every ocean basin, in every part of the world where organic matter is buried and the oxygen is gone. The methane produced this way carries a particular chemical signature. Deep in its molecular structure, in the ratio of carbon atoms it contains, is a record of its origin. The carbon in biologically produced methane is slightly lighter than average. Its atoms have one fewer neutron than the most common form, and so when this gas is recovered and analyzed, its carbon signature reads more negative than negative 60 parts per thousand, a value so depleted in heavy carbon that no geological process produces it. Only biology, working slowly in cold darkness, creates methane this light. It is, in a sense, a fingerprint of life, pressed into the chemistry of a gas that will eventually travel upward through hundreds of meters of sediment toward the ocean above. Not all methane in the deep sediment comes from living microbes. In regions where sedimentary layers are buried very deep and subjected to the sustained heat of the earth below, the organic matter itself can be cracked apart by temperature and pressure alone, breaking into shorter hydrocarbon chains without the help of any organism. This thermogenic methane, as it is called, forms from the same ancient organic material, but through a different process, a slow thermal breaking rather than a microbial digestion. It carries a heavier carbon signature, richer in the denser form of the atom, and it arrives accompanied by other gases, ethane, propane, butane, each a slightly longer hydrocarbon chain produced by the same deep cracking process. Where these heavier companions appear, they tell researchers something about the depth of burial and the heat that the source rock has endured, a chemical record of conditions far below anything that can be directly sampled. Both kinds of methane eventually begin to rise. The buoyancy of gas and fluid and the pressure of sediment compressing from above push the methane upward through the pore water of the sediment column. It diffuses slowly through the spaces between grains, travels along microscopic channels, and follows the path of least resistance toward the seafloor surface above. The journey takes time. In low-flux sediments, a single molecule might spend centuries working its way upward through a few meters of fine-grained mud. In higher flux regions, where faults or permeable layers provide a more direct pathway, the migration is faster. But in either case, somewhere along the way, something is waiting. Between the zone where methane rises and the zone where seawater sulfate slowly diffuses downward from the ocean above, there is a meeting place. It has no sharp physical boundary that the eye could see. The sediment looks the same on either side of it, dark, fine-grained, cold, compressed, but at this particular depth within the sediment column, two opposing chemical gradients collide: methane, moving upward from below, sulfate, moving downward from the ocean above. Where they meet, a chemical transformation occurs that controls the fate of nearly all the methane produced in these sediments. This meeting place is called the sulfate-methane transition zone, and its position below the seafloor shifts depending on local conditions. Deeper in low-flux sediments, where methane arrives slowly; shallower, where the upward flux is strong enough to push the boundary toward the surface. The chemistry that happens at this boundary is ancient and very nearly perfect in its efficiency. Two groups of microorganisms live here, so closely entangled that they have come to function as a single metabolic unit. One group is archaeal, among the most ancient forms of cellular life on Earth, belonging to a lineage that diverged from the common ancestor of all life before the oceans had their present composition. These are the anaerobic methane oxidizing archaea, called ANME for short. They carry out a process that is, in essence, the reverse of what their relatives in the shallower sediment do. Instead of making methane, they consume it. Instead of releasing energy by generating methane, they extract energy by carefully dismantling it. The amount of energy available from this reaction is very small. The archaea that perform it are working at one of the lowest energy yields of any known metabolism on Earth, capturing just enough to grow and divide, no more. Paired with the ANME archaea in tight physical contact are sulfate-reducing bacteria. These belong to a lineage of bacteria that also thrive without oxygen, using sulfate dissolved in seawater as their terminal electron acceptor, the chemical that accepts the electrons that other organisms would hand off to oxygen. Where a human cell passes its electrons to oxygen and produces water, these bacteria pass their electrons to sulfate and produce hydrogen sulfide. The partnership works because the archaea produce something the bacteria need, and the bacteria produce something the archaea need. The precise nature of what passes between them is still not fully settled. The best current picture involves a form of electron transfer across the physical junction between the two cell types, possibly through conductive protein filaments, so thin they fall below the resolution of ordinary light microscopes. Whatever the exact mechanism, the result is that together these two organisms do what neither can accomplish alone. They oxidize methane using sulfate in a reaction that couples two processes and shares the meager energy between them. The methane enters the consortium and is dismantled. The sulfate enters and is reduced. Hydrogen sulfide and dissolved carbon exit. And the methane molecule, which began its journey in the deep sediment and has been slowly rising toward the water column above, ends here, consumed in the cold dark, a few centimeters below the seafloor surface, where something else will use what remains. The efficiency of this process is extraordinary. Across cold seep environments globally, it is estimated that somewhere between 70 and 90% of all upward migrating methane is intercepted and consumed before it ever reaches the open water. The ocean floor, read from the outside, looks still and passive. Underneath it, a slow chemical filter is running continuously, processing methane at a rate that, summed across every seep and every centimeter of active sediment transition zone on Earth, amounts to tens of billions of kilograms of methane carbon consumed per year. The organisms doing this are invisible. They are smaller than the width of a human hair. Most of them will never be cultured in a laboratory, and yet they represent one of the most consequential metabolic processes in the ocean's carbon cycle. What they leave behind matters just as much as what they consume. Hydrogen sulfide, the byproduct of the coupled reaction, does not disappear. It begins its own journey upward through the sediment, diffusing toward the seafloor surface, just as the methane diffused from below. As it rises, it encounters the iron compounds present in the sediment minerals around it, and some fraction reacts immediately, binding with iron to form iron sulfide minerals, dark, fine-grained crystals that incorporate themselves into the sediment matrix and stay there. In thick, ancient seep sediments, these minerals accumulate in layers, recording the history of past sulfide production in the geochemical signature of the rock. A core pulled from such sediment and examined for its iron sulfide content reveals a time series of seep activity, intervals of intense flux and quiet, the rhythms of the Earth's interior expressed as mineral chemistry in dark mud. But not all the sulfide is captured this way. A fraction continues upward, escaping the sediment surface and entering the overlying water, where it serves as the primary energy currency for a community of life that depends on it entirely. This is the sulfide that reaches the chemosynthetic organisms living at and just above the seafloor, the fuel that drives an entire ecosystem in the permanent absence of sunlight. Without the slow, patient, nearly invisible work of the ANME consortia and the sulfate-reducing bacteria below, this fuel would not exist. The community above is fed by chemistry that takes place in darkness, centimeters underground, performed by organisms that require no light, no warmth, and almost no energy to survive. What the sulfide does not do on the way up, it does when it arrives. The alkalinity of the pore water also changes in the wake of the methane oxidation reaction. When methane is oxidized and sulfate is reduced, Bicarbonate ions accumulate in the pore water, the water trapped between sediment grains, and when bicarbonate concentrations climb high enough, carbonate minerals begin to precipitate. Calcium carbonate, drawn from the dissolved minerals in the surrounding seawater, begins crystallizing out of solution around the active seep, forming crusts and slabs and nodules of pale rock directly on and just below the seafloor surface. This process is slow. Individual mineral crystals form over years. Larger crusts develop over decades or centuries. But the result across the full lifespan of an active seep is a hard substrate, an irregular pavement of authigenic carbonate rock, pale and rough-surfaced, shaped entirely by the chemistry of methane oxidation rather than by any geological compression or volcanic heat. The word authigenic simply means formed in place. The carbonate grew where it now sits, precipitated from the chemistry of the seep itself, rather than deposited from elsewhere. These rocks bear the chemical memory of the reactions that made them. The carbon in the carbonate comes partly from the bicarbonate produced by methane oxidation, and so it carries the same light isotopic signature as the biogenic methane that was consumed. A fragment of authigenic carbonate, cracked open and analyzed, reveals its origin in the ratio of its carbon atoms, the same light signature, now locked permanently into stone. Where these rocks are found, whether on the modern seafloor or in ancient sedimentary formations exposed on land, they mark the places where methane seeps once ran, a durable and readable record of chemistry that played out millions of years ago in a world that looked very different from this one. At the seep surface, where the sulfide arrives and the carbonate is building, the chemistry continues. Hydrogen sulfide emerging from the sediment into the overlying water is quickly intercepted by another community of organisms, bacteria living at the sediment surface or just above it, capable of oxidizing sulfide back to elemental sulfur or sulfate using the oxygen that diffuses down from the open ocean above. These bacteria are not the centrophic consortia working in the sediment below. They are aerobic, or at least able to bridge the aerobic-anaerobic boundary, occupying the zone where sulfide from below meets oxygen from above. In this narrow chemical gradient, they find extraordinary abundance, more chemical energy available per unit volume than almost anywhere else in the deep ocean. The mats they form are thick, pale, and dense, visible to a remotely operated vehicle's cameras as white or orange fields on the seep surface, sometimes covering several square meters of seafloor in a continuous living layer. The sulfide also reacts in other ways. In the chemistry of cold seep sediment, where concentrations of dissolved sulfide can reach levels that would be immediately lethal to most organisms on Earth. Iron is not the only mineral being altered. The pore water itself is being transformed, its pH reduced slightly, its chemistry shifted away from the conditions of ordinary seawater towards something harsher, more reducing, more saturated with compounds that ordinary marine life cannot tolerate. The boundary between the seep and the surrounding seafloor is not just a biological boundary. It is a chemical boundary, a zone where the chemistry of the sediment and the water changes over a scale of centimeters, and where organisms must carry physiological adaptations that allow them to bridge the difference. Now, set against all of this, the rising methane, the consuming archaea, the producing sulfide, the precipitating carbonate. There is another form of stored methane that belongs in this picture, one that is neither gas dissolved in pore water, nor gas rising as free bubbles, but something else entirely. Under conditions of high pressure and low temperature, methane does not behave the way it does at the surface of the Earth. Instead of remaining a gas mixed into fluid, It can be incorporated into the crystalline lattice of water molecules, locked inside a molecular cage of hydrogen-bonded water in a solid structure that looks and feels like ice but is not ice. These structures are called methane hydrates, and they form naturally in the deep sea sediments where pressure is high enough and temperature is low enough to keep them stable, generally in water deeper than about 1,500 feet, though the exact depth varies with the local temperature of the water above. Within their zone of stability, methane hydrates are a remarkable thing to encounter. They are pale, almost luminous in the dark, cold, smooth, with the visual texture of dense ice, sometimes forming mounds or outcrops on the seafloor, where methane flux is high enough to have built them up over time. At the surface, a fragment brought up rapidly will begin to fizz and dissociate, releasing methane gas as the pressure drops and the crystal structure collapses. But on the deep sea floor, in the right conditions, they simply persist, stable, quiet, holding methane in solid form in quantities that are, globally, very large. The total methane stored in seafloor hydrates worldwide is estimated to be a significant fraction of all the carbon in the atmosphere. Though the precise figure is difficult to measure, and the estimates have changed considerably as survey methods have improved. What makes hydrates consequential is not just the methane they hold, it is the conditions under which they release it. The stability of a methane hydrate depends on the precise combination of pressure and temperature at the point where it sits. Pressure is set primarily by the depth of the water above. It does not change on human timescales unless the ocean itself is somehow draining. But temperature is different. If the water at the seafloor warms, even slightly, the zone of hydrate stability shifts. Hydrates that sit just within the lower boundary of that zone can become unstable, their crystal lattice weakening and eventually dissociating, releasing the methane they contain into the pore water and potentially into the overlying ocean. This dissociation happens gradually, at rates set by how quickly heat diffuses into cold sediment. It is not, under most natural conditions, an explosive process. It is slow, patient, and continuous, like most of the chemistry in these sediments, but over long enough timescales, even slow dissociation can move very large quantities of methane. The methane released by dissociating hydrates joins the upward flux already present in the Seeps system, adding to the supply that the ANME consortia must process, and that, if the flux is high enough, may escape to the water column as free gas. In some seep regions, hydrate dissociation has been observed to drive particularly vigorous bubble streams, rising from the seafloor, columns of methane gas moving upward through the water column, some rising hundreds of meters before dissolving into the surrounding seawater. Most of this dissolved methane is consumed by bacteria in the water column before it reaches the surface. The ocean, like the sediment, runs its own methane filter at multiple levels. But the hydrate is also a kind of cap, a partial lid on the methane below. In regions where hydrates are thick and continuous, they can reduce the upward migration of gas, holding it in place and building up pressure in the sediment beneath. When that cap is breached by a submarine landslide, by a sudden pressure change, by a shift in the thermal structure of the overlying water, the release can be more concentrated and rapid than the background seep process produces. Craters on the seafloor in some regions are thought to have formed by exactly this mechanism, ancient explosions of gas-saturated sediment that left behind depressions still visible today in detailed surveys of the ocean floor. What sits at the surface of all this chemistry, at the top of the sediment, at the boundary between the floor and the ocean, is a world that draws its entire energy budget from processes happening in the dark below. No sunlight reaches here. No photosynthesis is possible. The energy that drives the organisms living at a cold seep comes from the chemical gradient between reduced compounds rising from below and oxidized compounds, primarily oxygen and sulfate, supplied by the ocean above. The steeper that gradient, the more energy is available. The more energy that is available, the denser and more diverse the community that the seep can support. The sulfate methane transition zone, for all that it lies hidden in the sediment, is the foundation. It is the place where the chemical energy of buried organic matter, transformed over time into methane and then intercepted and partially converted into sulfide, is finally made available to larger and larger organisms moving up the chain of the food web. The archaea and bacteria in the sediment are the first step of this chain. They are the only step that runs entirely on chemistry, with no biological intermediary above them. Everything else at the seep, every organism visible to a camera, every shell attached to a carbonate slab, every animal moving across the soft sediment at the edge of the active zone exists because of what is happening in the dark below, in the cold, in the chemical interface that has no name. A person would recognize without a field guide and no beauty that any eye could see. The chemistry is the world. Here, everything else is built on it. Deep in the sediment, the pore water continues to carry its cargo of dissolved compounds, sulfide rising, sulfate sinking, methane pressing upward through grain and fracture, and the thin aqueous films between particles too fine to see. The transition zone shifts slightly as fluid flux changes, deepening over decades when the seep quiets, shoaling when activity intensifies. Authigenic carbonate grows in the sediment around it, hardening the mud into stone so slowly that a researcher measuring the same outcrop in 2 successive years would find no detectable difference. The ANME consortia in the sediment above process methane at whatever rate the flux provides, neither faster nor slower, with no response to anything except chemistry. There is something in all of this that is very old. The reactions taking place in cold seep sediments today are not recent inventions of life. The metabolic pathways involved, the archaeal reversal of methanogenesis, the bacterial reduction of sulfate, belong to some of the deepest branches of the tree of life. They are ancient processes, running in conditions that resemble the early ocean more closely than anything at the sunlit surface. The microorganisms performing them are not highly evolved specialists shaped by the complex pressures of a diverse ecosystem. They are survivors of the earliest chemistry of life on Earth, continuing to do what their ancestors did in an ocean that looked nothing like today's, in a world where the atmosphere had no oxygen and all of life was microbial, and the sea floor was the only place that anything happened at all. The methane in the sediment carries the carbon of ancient organisms. The microbes that consume it carry metabolic strategies older than complex life. The chemistry they enact is patient and slow and deeply repeated across every ocean floor, where organic matter has been buried and pressure and time have done their work. It runs without light, without seasons, without any signal from the world above. The only clock it follows is the slow diffusion of chemicals through porous sediment, a timer set in centimeters per year rather than in hours, a tempo so slow that measured against a human life, it appears to stand completely still. On the seafloor above the sediment, the pale outcrops of authigenic carbonate rest exactly where they formed, shaped by the chemistry that has been building them for longer than any record keeps. Cold water moves across them in slow, deep currents. No light reaches them. No sound of the surface world arrives here. Not the wind or the waves or anything carried by air. There is only the pressure of a kilometer of water above, and the cold, and the slow release of chemistry from below, sulfide rising through sediment, methane dissolving into still dark water, the seep continuing in the same way it has continued since long before anything looked at it, and in the same way it will continue long after this narration is done. The methane moves upward through the cold. The crystals of hydrate hold their methane in silence in the dark. The transition zone sits at its particular depth within the sediment, processing chemistry at the rate the flux permits, unchanged and unchanging, a chemical boundary in a column of dark sediment at the bottom of a cold ocean, held in its delicate equilibrium by temperature and pressure and the slow, ceaseless movement of molecules through the deep. Somewhere deep in the cold sediment of the continental margin, smaller than the period at the end of this sentence, a cluster of cells sits in complete and permanent darkness. No light has ever reached this place. No current stirs the pore water, filling the narrow spaces between sediment grains. The temperature is only a degree or two above the freezing point of seawater, and has been so for as long as the sediment has lain here. And yet, inside this cluster of cells, a paired sphere of archaea and bacteria so small that several thousand of them together would not span the width of a human hair. Something is happening. Something has been happening continuously for as long as this sediment has been saturated with methane rising from below. One partner in the cluster reaches into the surrounding pore water and intercepts a molecule of methane. Through a chain of biochemical steps that researchers are still working to fully understand, it begins to run that molecule's chemistry in reverse. The reaction produces almost no energy, it yields almost nothing, and yet it is one of the most consequential acts performed anywhere on Earth. These paired clusters are called ANME consortia. The first part of the name comes from their role, anaerobic methanotrophic archaea, the ancient single-celled organisms whose metabolic partnership with sulfate-reducing bacteria makes them the primary biological filter between the methane stored beneath the seafloor and the ocean water above. They are not rare. Across every cold seep, on every continental margin, through sediments on the slopes of ocean basins from the poles to the tropics, these consortia exist in densities that can reach hundreds of millions per cubic centimeter of sediment. They are among the most abundant specialized microbial forms on Earth. They are also invisible to the unaided eye and have been performing their chemistry without interruption since long before there were instruments to detect them. 3 broad groups of anaerobic methanotrophic archaea have been identified, and they are not identical in their habits. The first group, called ANME1, tends to inhabit deeper sediment layers, where methane flux is lower and more diffuse, where the chemical gradients are gentle and the pace of activity slower. And M1 cells sometimes live without being physically attached to their bacterial partners, existing in a looser metabolic arrangement whose precise mechanism still holds open questions. The second and third groups, NME2 and NME3, are more commonly found in higher flux environments, closer to the seafloor surface, where methane arrives faster and the chemistry is sharper. These are the groups most often seen forming tight, physically intimate consortia with sulfate-reducing bacteria, the 2 cell types pressed together in dense spherical aggregates, the archaeal cells typically forming a core surrounded by a shell of bacterial cells. Or sometimes the arrangement is reversed, or sometimes it is more irregular. The exact geometry varies, and not all of it is yet fully explained. The bacterial partners most commonly found, paired with ANE archaea, belong to a group called Desulfosarcina and Desulfococcus, both members of a broader bacterial division whose central metabolic role is the reduction of sulfate. In the cold pore water of seep sediment, these bacteria take the sulfate that diffuses down from seawater above and use it to accept the electrons that their archaeal partners pass to them. The result of this exchange, the output of the entire consortium's shared metabolism, is hydrogen sulfide. The sulfide diffuses upward through the sediment. It is, to most life on Earth, a lethal compound, capable of disrupting cellular respiration at concentrations far lower than what seep sediment routinely contains. But in the context of the cold seep, it is fuel. It is the currency that flows from the microbial world deep in the sediment upward toward the water column, where it will become the energetic foundation for an entire ecosystem. The mechanism by which the ANE archaea and their bacterial partners exchange energy across their shared membrane, or the narrow space between their cells, has been a subject of careful research for years. One hypothesis holds that direct interspecies electron transfer occurs through nanowire-like filaments, conductive structures bridging the two cell types and allowing electrons to pass without any chemical carrier molecule in between. There is evidence for this. There are images from electron microscopes showing fine filamentous connections between paired cells and measurements of electrical conductance in intact consortia. The evidence is genuine, and the picture is becoming clearer, though not all details have closed. What is certain is that the two cell types work together, that neither performs well alone in this environment, and that their combined action is responsible for consuming somewhere between 70 and 90% of all the methane that migrates upward through seep sediment before it ever reaches the open water. That figure deserves a moment of quiet attention. Deep in the sediment, in the cold and in the dark, in clusters too small to see with the naked eye, these organisms intercept and consume the vast majority of the methane that the geology of the seafloor produces and drives upward. The methane that bubbles from the sediment surface, the wisps that rise through the water column and occasionally reach the atmosphere, that is what escapes the filter. That is the fraction that gets through. The rest is processed silently below. The depth at which this microbial processing is most concentrated is not fixed. It shifts in any given column of sediment at a cold seep. There is a horizon, a layer, where the chemistry of two opposing flows converges. From above, sulfate dissolved in seawater diffuses downward through the sediment pore water. From below, methane carried in pore water diffuses upward. The two compounds move toward each other through the sediment, their concentration gradients opposing, and at the depth where they meet in roughly equal supply, the anaerobic methanotrophic consortia are densest. This horizon is called the sulfate-methane transition zone. It is not a visible line. It has no color, no texture, no edge that an instrument could touch. It is a chemical fact, a depth within the sediment where one gradient overtakes the other, and it can lie anywhere from a few centimeters below the seafloor surface in areas of very high methane flux to several meters deep in areas where methane seeps slowly and dilutely. Where the methane arrives fast, the transition zone is shallow and sharp, and the consortia are tightly packed within it. Where the methane arrives slowly, the zone sinks deeper into the sediment, and the microbial community within it is more diffuse. The depth of that invisible horizon is a direct reading of how much methane the geology below is sending upward. Within the transition zone, the microbial consortia are not merely present. They are dense enough in the most active seep cores to collectively respire at rates that have been measured using radio tracers, molecules with detectable isotopic labels introduced into sediment cores and tracked as they are consumed. In the quieter peripheral zones of a seep, where methane is dilute and flux is slow, The rate of methane consumption may be less than 1 nanomole per cubic centimeter per day, a quantity so small it takes specialized instruments to confirm it is happening at all. At the most intensely active seep centers, in sediment directly above the highest flux fluid conduits, measured rates have exceeded 1,000 nanomoles per cubic centimeter per day. The difference between the quietest and most active zones spans more than 3 orders of magnitude, contained within a few meters of lateral distance across the seafloor surface. Summed across all of the cold seeps on Earth's continental margins, the annual methane consumption by anaerobic methanotrophic communities has been estimated at somewhere between 60 and 300 teragrams of methane carbon per year. A teragram is a trillion grams. Even the lower end of that range represents an immense quantity of a potent greenhouse gas processed by organisms no one had ever seen or suspected until the late 20th century, operating in complete darkness at the bottom of the sea. They do not do it intentionally. They do it because they are metabolically suited to it, and because the chemistry of their environment makes it possible, and because they have been doing it in various forms for what are almost certainly hundreds of millions of years. From the microbial consortia in the transition zone, the hydrogen sulfide they produce begins its slow upward movement through the sediment pore water. As it rises, it enters a different chemical regime, one closer to the seafloor surface and closer to the oxygen that diffuses down from the overlying water, where sulfide and oxygen meet, or in some cases where sulfide and nitrate meet, since some of these organisms store nitrate internally as an alternative oxidant. A different set of microbial metabolisms takes over. These are the sulfur-oxidizing bacteria. Unlike the ANME consortia, which are invisible within the sediment and detectable only through chemical measurement or microscopy, some of the sulfur-oxidizing bacteria at the seafloor surface form structures that can be seen with the unaided eye, or at the very least with the cameras of a remotely operated vehicle moving slowly across the seafloor. The most striking of these is a genus called Beggiatoa. A single Beggiatoa cell is large by bacterial standards. Some filaments reach over 100 micrometers in diameter, wider than many human cells, and measurable fractions of a millimeter across. The filaments grow end to end in long chains, each chain gliding slowly across the sediment surface, or lying densely matted with thousands of others, their collective mass forming the thick white coverings that are one of the most immediately recognizable signs of an active cold seep. From the cameras of a vehicle descending to a seep site, these mats appear as pale patches against the dark sediment, brilliant white in places, shading toward cream or gray at their edges, sometimes covering areas of several square meters around a vent opening. Inside each Beggiata filament, the cell biology is unusual even by bacterial standards. The cells are large enough to maintain a central vacuole, a fluid-filled internal compartment, and many Beggiata at cold seeps store nitrate within these vacuoles in concentrations far higher than the surrounding seawater. The cell, in effect, carries its own reservoir of oxidant internally, allowing it to function even when oxygen in the overlying water is seasonally low or locally depleted. The cells also store elemental sulfur as bright intracellular granules, visible under a microscope as tiny white or pale yellow spheres scattered through the cytoplasm. The sulfur granules are a reserve, partially oxidized sulfur compounds held within the cell, available to be processed further when conditions allow. A filament actively metabolizing sulfide, accumulating granules, gliding through a thin film of sulfide-rich water at the sediment surface, carries within it a chemical record of what it has been doing and what it has available to continue doing. The mat of Beggiatoa is not a uniform structure. Seen up close, it has texture and depth. The upper surface, in contact with the overlying water, is where oxygen or nitrate from that water is available. The lower surface is in contact with the sulfide-rich sediment, pore water below. Each filament tilts itself in effect to span this chemical gradient, its lower end immersed in sulfide, its upper end in oxidant. The mat as a whole is an enormous collective enzyme, a living interface between two chemical worlds, processing the sulfide that the invisible microbial consortia below have produced and sending sulfate back toward the sediment in a cycle that feeds back in small part to the consortia below. It is a tightly interlocked chemistry, and the mat is one of its most visible expressions. Two other sulfur oxidizing genera are commonly encountered at active seep surfaces. Theoploca forms bundles of long filaments enclosed within a shared sheath, a gelatinous tube that the filaments glide through vertically, their tips reaching into the sediment below and into the water above, shuttling sulfide upward and nitrate downward in a way that lets them exploit chemical gradients too steep for simpler organisms. Archobacter is a genus whose filaments are finer and more delicate, forming softer, sometimes translucent mats at seep sites, where fluid flow is somewhat gentler. At the edges of a highly active Beggiato field, where the thick white mat gives way to patchier coverage, Archobacter communities often fill the intermediate zone. The transition from one mat type to another across a few meters of seafloor is a map of the chemical conditions below. The flux rates, the sulfide concentrations, the availability of competing oxidants, written in the colors and textures of living microbial film. Not all seep mats are white. At some sites, particularly at lower flux margins or at the edges of active zones, the mats take on an orange or pale rust color. This color reflects sulfur compounds in an intermediate oxidation state, not fully reduced sulfate, not fully oxidized sulfate, but something partway between, held in that state by the balance of supply and demand at that particular patch of seafloor. An orange mat is a quiet mat. The chemistry beneath it is slower, the sulfide arriving more dilutely, the microbial community thinner and more widely spaced. The color is not decoration. It is a chemical signal about what is happening in the sediment underneath, readable by those who know how to look, even in the total darkness of the deep sea, lit only by instrument light. There is a tendency, when describing microbial communities, to treat them as uniform, as carpets or clouds of identical cells doing identical things. The reality in seep sediment is more complex and more interesting than any simple picture allows. Along any vertical centimeter of active seep sediment, the microbial community changes. Near the surface, where oxygen or nitrate is available, sulfur oxidizing bacteria dominate. A centimeter or two lower, the oxygen is exhausted, and the community shifts toward anaerobic metabolisms. At the sulfate-methane transition zone, the ANME consortia are densest. Below that, in sediment where sulfide is nearly gone and methane is abundant, other archaeolineages, methanogens, organisms that produce methane rather than consuming it, may be present, adding yet another metabolic layer to the vertical stack. The sediment is not a uniform matrix. It is a column of distinct chemical zones, each hosting its own community, each community shaped by the precise mixture of compounds available at that exact depth. The boundaries between these zones are not walls, they are gradients. A given cell type does not stop abruptly at a chemical threshold and hand off to the next community below it. There is overlap, competition, and in some cases cooperation across the boundaries. The AME archaea at the transition zone exist within a few millimeters of the sulfur oxidizing bacteria above them and within a few millimeters of the methanogens below. This vertical compression of metabolic diversity, the stacking of fundamentally different energy-yielding chemistries within a column of sediment no deeper than a finger is long, is one of the features of Seeps sediment that makes it one of the most metabolically rich environments anywhere in the biosphere. Among the most carefully studied aspects of seep microbiology is the question of how much energy the AME consortia are actually obtaining from their metabolism. Anaerobic oxidation of methane, coupled to sulfate reduction, releases very little free energy, roughly 1/16th of what aerobic respiration of the same amount of organic matter would yield. The consortia are operating energetically on the minimum margin. They grow slowly. They divide rarely. A single ANME consortium may take months to double its biomass under the conditions of a typical seep, and yet they persist in enormous numbers over geological time. The extreme energetic austerity of their lifestyle is not a flaw or a limitation that evolution has failed to overcome. It is a match to the environment. The chemistry of their setting provides very little energy, and they have specialized over deep time to extract enough from that little to survive and to maintain the community that has been filtering the seafloor's methane for longer than most rock formations have existed. Because their growth is slow, the turnover of the ANME community in any given sediment column is also slow. Cells that die are replaced by the division of cells that remain, but the replacement is gradual. This means that the community in a given volume of sediment has in some sense been accumulating over a long time, not the same individual cells, but the same lineages, performing the same chemistry, in sediment whose oldest layers were deposited centuries or millennia ago. The consortium sitting in the sediment now is the descendant of a consortium that sat there when the overlying ocean was a different temperature, when different species of whale swam above it, when the coastlines above the continental margin looked slightly different. The chemistry has not changed. The methane still rises. The consortia still intercept it. The sulfate-methane transition zone still sits at whatever depth the flux rates currently dictate. The sulfide produced by all of this microbial activity does not remain neatly in the pore water. Some of it reacts with iron in the sediment, forming pyrite and other iron sulfide minerals that are stable solids under seep conditions. These mineral precipitates accumulate in the sediment over time, creating a geochemical record of past seep activity that can persist long after the fluid flow has shifted or stopped. A sediment core extracted from a former seep site may contain layers of iron sulfide minerals that mark past high flux intervals, even when the current chemistry of the pore water shows little sign of active seepage. In this way, the microbial community leaves a mineral record of its activity, a signature in the sediment that persists on timescales far longer than the cells themselves. The mat-forming bacteria at the seafloor surface also participate in mineral cycling, though in different ways. Beggiato communities have been associated with changes in the oxidation state of manganese and iron at the sediment surface, and their activity influences the precipitation and dissolution of metal compounds in the uppermost sediment layer. The mat, in effect, is not simply sitting on the sediment, it is chemically altering it, changing its mineral composition through the ongoing flux of electrons passing through its cells. The sediment under a thick Beggiato mat looks chemically different from sediment an arm's length away, outside the mat's boundary. The difference is written in the minerals, in the iron and manganese and sulfur compounds, in the ratio of oxidized to reduced forms of each element, and it reflects the sustained metabolic activity of the mat community above. The question of how these microbial communities first established themselves at a new seep site is one that research has begun to illuminate, but has not fully resolved. When a new fluid pathway opens in the sea floor, when a fault shifts slightly and allows methane-rich pore water to find a new route upward, the sediment at the surface changes. The pore water chemistry shifts. Sulfide begins to appear where it was not present before. The AME consortia, already present in the sediment at low densities throughout large areas of the deep sea floor, do not need to arrive from elsewhere. They are already there, at low abundance, living on trace amounts of methane in normal deep sea sediment. When the fluid flux increases, conditions become more favorable, and the consortium population grows slowly toward the higher densities characteristic of active seep centers. The mat-forming bacteria at the surface respond similarly. Some are present in background seawater and sediment, capable of colonizing a new sulfide-rich surface when one becomes available. The pace of this establishment is slow, matched to the pace of the organisms themselves. A new seep does not erupt into biological activity overnight. It accumulates it over months and years as the microbial communities develop from background to mat-forming densities in response to the shifting chemistry. The first signs of biological activity at a new seep site are often chemical rather than visual. The sulfide plume in the water above the vent, detectable by sensors before any mat is visible. The mat itself comes later, as the community builds sufficient biomass to form a visible layer, and later still, the larger organisms, the mussels, the clams, the tube worms arrive, drawn in part by the chemical signals, and in part simply by the sulfide-rich environment the microbial community has created. The foundation is always microbial. The visible community that eventually develops above it, and the remarkable animals that inhabit it, rest upon this invisible, ancient, chemically driven base. Across the floors of Earth's continental margins, in sediment pressed down by kilometers of cold ocean water, in absolute darkness and near freezing cold, these communities persist. They are the base of an ecosystem that extends upward from the sediment, through the mat, through the water, just above the vent, through the bodies of the larger organisms that settle and grow at the seep surface. But the base itself, the ANME consortia, deep in the transition zone, the sulfur-oxidizing bacteria at the surface, the slow cycling of sulfide and sulfate and iron and methane through the sediment column, is its own world, complete in itself, requiring nothing from sunlight, nothing from the atmosphere, nothing from the surface ocean, a kilometer or more above. It requires only the methane rising from below, and the sulfate diffusing down from the seawater above, and the pressure of deep time pressing those two gradients together across a chemical horizon that no instrument can see, but that every cell within it can feel. The Beggiato filaments lie across the dark sediment in long pale curves. Each filament is a chain of cells, each cell gliding slowly along the surface or into the sediment below, each cell storing within it tiny bright granules of sulfur that it has been accumulating from the sulfide-rich water around it. In the stillness of the deep sea floor, nothing moves quickly. The filaments are not still. They glide, by mechanisms that are not fully explained, at rates so slow that the motion is invisible in real time. But they move. They reorient to follow chemical gradients. They slide across the sediment surface, following the invisible traces of sulfide concentration, finding the richest zones and covering them. The mat is not passive. It is moving, slowly, in the dark, following the chemistry that sustains it. Somewhere in the sediment, a centimeter below the mat surface, An ANME consortium is intercepting methane. The archaeal core of the consortium reaches its chemistry into the surrounding pore water. The electron transfer passes to the bacterial shell. Sulfate arrives from above. Hydrogen sulfide forms and diffuses away, upward, toward the mat, toward the transition zone boundary, toward the larger world that the chemistry of this consortium is quietly helping to build. The consortium will continue for months before it divides. The cell lineage within it has been continuing for much longer than that. Neither the archaea nor the bacteria know what they are part of. They process what arrives, yield what their chemistry produces, and persist. In the settled dark of the deep sediment, where no light has ever penetrated and no current has ever reached, this has been enough. It has been enough for geological time, and the methane that rises from below, and the sulfate that sinks from above, and the chemical horizon where they meet and react. These gradients will persist long after every instrument that has ever measured them has rusted and dissolved. The consortia within the transition zone will still be there in some form, processing what arrives. The mats will still be forming at the surface of active seeps, pale and slow and precise. The sulfide will still be rising from the sediment into the cold, dark water above, where it will become the first chemical link in a chain of life that builds itself quietly and completely without the sun. Across the whole of the deep ocean floor, across every continental margin of every ocean basin, in water ranging from a few 100 feet deep to depths where the pressure would crush any unprotected object to nothing, these microbial communities are present. In sediment, wherever methane rises and sulfate descends, performing the same chemistry they have performed for hundreds of millions of years. Each individual consortium is too small to see. Each mat, by the standards of the surface world, covers a modest patch of ground, but taken together as a global community of organisms performing a shared metabolic function across the full extent of Earth's seafloor margins, they constitute one of the most expansive living systems on the planet, ancient, largely invisible, and deeply interwoven with the chemistry of the ocean itself. The filaments lie still in the cold. In the sediment below, the consortia process what arrives. The sulfide rises, the sulfate descends, and at the depth where these two flows meet, in the permanent and total dark of the transition zone, the oldest methane filtering communities on Earth continue the work that was their inheritance before the first animal had ever formed a mouth. In the cold sediment of the deep seafloor, something is living without a mouth. It has no stomach. It has no gut, no digesting chamber, no way to swallow anything at all. It is 2 meters long, anchored in the sediment below, and extending upward into dark water above. And it has never in its entire adult life consumed a single morsel of food in the ordinary sense. Its outer surface is a white tube of stiffened protein, smooth and slightly ridged along its length. From the upper end, where the tube opens to the water, a cluster of filaments extends outward, dense and red, the deep red of blood spreading into the cold like the petals of something that has never known a sun. This is the vestimentiferan tubeworm, and the life it lives in that cold, dark place several 100 feet below the surface of the sea is built entirely from the inside. Within the body of this worm, where a gut would be in most animals, there is instead a dense organ packed with bacteria. It fills most of the interior. It is called the trophosome, from roots meaning nourishment and body, and it is precisely that, a body whose function is to hold the organisms that feed the organism holding them. The bacteria inside the trophosome are sulfur-oxidizing chemosynthetic cells, each one fixing carbon from the surrounding water and releasing organic compounds that diffuse directly into the worm's tissues. The worm does not eat the bacteria. The bacteria do not eat the worm. They are woven together, one inside the other, in a relationship that is held across geological time. The arrangement works because of a very particular piece of chemistry. Deep in the sediment below the worm's anchor point, hydrogen sulfide is rising through the pore water, carried upward from the horizon where methane and sulfate meet and react. The worm has extended root-like posterior structures downward into the sediment, thin tubes of tissue reaching toward that sulfide. It collects the sulfide there. At the same time, it draws oxygenated water across the red plume above. Both the sulfide and the oxygen are bound by a specialized molecule in the blood, a form of hemoglobin, unlike the kind found in most animals, capable of holding onto sulfide and oxygen simultaneously without allowing the two to react with each other. The hemoglobin carries them to the trophosomes separately, each to the bacteria waiting there, and the bacteria do the rest. The red of those plumes is the hemoglobin. It is the same color for the same reason as the red inside any living creature with iron in its blood. Deep in the ocean, in the dark, that color means the worm is running oxygen and sulfide through itself at the same time, a molecular balancing act performed continuously, day after unlit day, for a life that may last over a century. What feeds the tube worm feeds the mussel differently. Against the carbonate rock that builds up near the Seeps center, where authigenic mineral has hardened the sediment into pale rough slabs, there are beds of mussels. They press together in thousands, dark shells overlapping and stacked, filling every surface. The individual shell is smooth and elliptical, darkening toward the hinge and paler at the edges, and inside each one, the gill tissue is not the simple filtering structure found in a coastal mussel. It is dense and layered and occupied. Two entirely different populations of bacteria live within the cells of those gills. One population oxidizes hydrogen sulfide. The other oxidizes methane. The mussel does not choose between them. It houses both and draws on each according to what the local fluid chemistry offers. This double endosymbiosis is the mussel's great flexibility. Near the center of the seep, where methane bubbles still emerge, and the methane concentration in the pore water is high, the methane oxidizing bacteria in the gill are more active. Toward the edges of the seep, where sulfide diffuses up from the sediment in greater relative concentration, and methane flux has diminished, the sulfur oxidizing bacteria take the larger share of the metabolic work. The mussel itself is essentially passive in this management. Its gill tissue provides the bacteria with a housed, regulated, oxygen-supplied environment, and the bacteria provide the mussel with fixed organic carbon. The balance shifts on its own, tracking the chemistry of the water. Each dark shell is, in this way, a small and living instrument of the local seafloor chemistry, reading it continuously and responding without nerve or intention. A bed of these mussels can persist for decades. The shells accumulate layer by layer as individuals die and new ones settle. The dead shells become substrate for other organisms. Snails, small worms, amphipods, and the living ones continue producing organic carbon, some of which drifts away from the seep as particles, entering the water column above and becoming part of the slow nutritional rain that feeds the wider deep sea floor. From outside, the mussel bed looks still. It is not still. Inside each shell, in the gill cells, in the bacteria nested within those cells, chemistry is running continuously, quietly, at the same cold temperature the water has maintained for 1,000 years. Beneath those mussels, beneath the slabs of carbonate rock, beneath the microbial mats that coat the sediment, in white and faint orange. The clams have their own arrangement. Vesicomiid clams are large and heavy-shelled, pale as old bone, and they live with one end of their soft body pointed downward into the sediment and the other extended upward into the water above. The foot, thick and muscular, capable of slow extension and retraction, pushes down through the sediment to reach the sulfide-rich pore water below the sediment surface. The gills above draw oxygenated seawater across. The clam bridges the interface. It lives precisely at the boundary where two chemical worlds meet. The bacteria in the vesicomiid clam's gills are exclusively sulfur oxidizers. There is no methane oxidizing partner here, no double arrangement. These bacteria live inside specialized cells called bacteriocytes that fill the gill tissue, each bacteriocyte holding many bacterial cells within it, the bacteria drawing the sulfide that the foot collects below and producing organic carbon that the clam's tissues absorb. The gill itself has become a chemosynthetic organ, so enlarged by this function that it fills much of the interior of the clam's shell, crowding the other organs to the margins. There is something else about the vesicomiid clam's symbiosis that carries its own kind of weight. The bacteria are not acquired anew by each generation. They are transmitted vertically, mother to offspring, through the egg itself. When a female vesicomiid clam produces eggs, a small population of the symbiotic bacteria is incorporated into each egg during its formation. The offspring hatch already containing their symbionts. They do not find bacteria in the environment and pick them up. They carry them from the beginning, inherited like any other trait, passed through the same channel as every other feature of the animal's biology. The bacterial lineage and the clam lineage have been traveling together in this way for a very long time, and the two have become so deeply intertwined that the clam cannot survive without the bacteria, and the bacteria are found nowhere in the world outside the cells of their host. In the sediment beyond the clam beds and mussel fields where the seep's influence fades and the chemical gradients grow weaker, much smaller worms live in tubes so fine they are nearly invisible. These are frenulate saboglinid polychaetes, relatives of the large vestimentiferan tubeworms, but built on a different scale entirely, a few centimeters long and thinner than a thread. They live in glass-like transparent tubes they construct in the sediment, and they too carry chemosynthetic bacteria. Their trophosome is a small structure proportional to their size, and the sulfide concentrations they depend on are lower than those at the seep center. In the peripheral zones, where the transition between seep and open deep-sea floor is gradual, and the chemical signals from below are faint, these small worms extend the seep community outward into territory that would otherwise carry no sign of the chemistry below. Among these relatives there is one genus, called Sclerolinum, that appears not only at cold seeps, but at other places on the deep sea floor, where organic matter accumulates and begins to decay under low oxygen conditions, sunken whale carcasses, waterlogged wood on the bottom, the decaying remains of any large organic mass that has sunk from above. Sclerolinum is a generalist in the world of deep-sea reducing environments, making its living wherever sulfide can be found, treating the cold seep and the decomposing whale as variations of the same chemical opportunity. Its presence in so many types of deep-sea habitat suggests that the physiological arrangement of housing chemosynthetic bacteria inside a saboglinid worm is ancient and adaptable, shaped long ago and applied widely. On the surface of the white beggiato mats that cover the most active seep sediment, something else moves. It is small and pale and carries a thick shell. Provana is a genus of gastropod snail found commonly at cold seeps, its shell heavily mineralized, denser and more solid than a typical snail shell of similar size. The shells of seep-dwelling snails live in water with elevated sulfide concentrations and locally reduced pH, the poor water chemistry altering the water just above the sediment surface in ways that slowly attack calcium carbonate. A thicker, more heavily built shell resists that dissolution. The Pravana snail moves across the mat surface on its muscular foot, rasping at the bacterial filaments with a toothed structure called a radula. It grazes the mat. It takes the bacteria directly, extracting their organic carbon the old-fashioned way, eating. The Pravana snail is not chemosymbiotic. It does not carry bacteria in its tissues. It simply eats the bacteria that other organisms depend on, feeding at the base of the food web that chemosynthesis has built. In this, it is ecologically ordinary, a grazer in a garden made of sulfur and methane, yet the shell it carries, pale and thick in the faint instrument light of a passing submersible, is itself a record of adaptation. Its weight and density are a response to the chemistry of the seep. The animal's body has been shaped by the environment it grazes, just as any grazer on the land is shaped by the roughness and toxicity of the plants it eats. Moving across the edges of a seep site, often in groups, are the galatothade crabs, small crustaceans with pale, almost white bodies, their movements deliberate and slow in the cold water. They are not chemosymbiotic either. They are scavengers and opportunists. A dead mussel at the seep edge, its shell cracked open by the slow movement of carbonate rock or by the pressure of a feeding fish, draws them in. Several will arrive and work at the exposed tissue together, their small chelae picking at the soft mass with a methodical patience. They graze at the margins of microbial mats. They take what is available at the edges of the seep community, and in eating, they transfer the seep's chemosynthetic carbon into their own bodies, from which it may eventually pass to the fish and cephalopods that visit from the surrounding deep-sea floor. The crabs are visible. They can be seen in ROV footage, moving through the light, pale bodies against dark sediment, claws working slowly. In their visibility and their mobility, they are the most legible part of the seep ecosystem to any observer. The animals that move, that approach the camera, that respond to disturbance, but they are the farthest from the chemistry that runs the whole system. They live at the edge of it, benefiting from what has been built below and within and between, arriving at the output of an enormous invisible process to collect what has drifted to the surface. The invisible process begins in the trophosome. It begins in the bacteria site of the clam's gill. It begins in the dual-occupied tissue of the mussel, where two bacterial populations divide the chemical work of the water between them. These inner arrangements, bodies built around bacteria built into bodies, are what make the seep community possible. Everything else builds from them. Consider what it means for an animal to have no digestive system. The absence of a gut in the adult vestimentiferan tubeworm is not a loss. It is an arrival at a different solution to the problem of being alive. The ancestral worm had a gut. At some point in deep evolutionary time, the lineage that would become the suboglonid worms began to carry bacteria in their tissues, and over immense spans of time, the external digestion became unnecessary, then vestigial, then absent. The trophosome expanded to fill the space the gut had occupied. The worm that emerged from this long process was not a lesser version of an animal that could eat. It was an animal whose interior had been reorganized around a different energy source entirely. An animal that does not eat the world, but contracts with a part of it, shelters it, and lives on what that part produces. The reorganization is visible in the blood. That specialized hemoglobin, capable of binding sulfide without allowing it to react with oxygen, is unique to these worms. Nothing else has quite this molecule, this specific adaptation to the problem of carrying two reactive compounds through the same bloodstream without catastrophe. The molecule is the record of the problem it solved, the problem of getting sulfide from the sediment to the bacteria without poisoning the animal's own tissues along the way. Inside every tube worm alive, at every seep and every ocean basin, that molecule is circulating, doing the same work it was shaped to do across millions of years. The mussels solve the same problem differently. Instead of routing sulfide through a blood molecule, the Bathymotomous mussel concentrates both sulfide and methane in its gill tissue, where the bacteria are already waiting. The gill is the interface. Oxygenated water moves over it from outside, and the gill is pressed against the seep-influenced water near the sea floor, where sulfide and methane are elevated. The bacteria at the gill's surface encounter both compounds directly. The mussel's body is the architecture that brings the compounds and the bacteria together in the same small space, with oxygen on one side and reducing compounds on the other. Two different solutions, two different animal bodies. The same fundamental task delivering chemical energy to bacteria housed within, accomplished by two lineages that arrived at the seep through different evolutionary histories and built different structures to manage the same chemistry. The clam solution is the most spatially dramatic. The foot goes down. The siphon draws water from above. The clam's body is a passage between two chemical environments, a biological bridge crossing the sediment surface, and everything about its anatomy, the shape of the foot, the size of the gill, the capacity of the circulatory system, reflects the demands of that crossing. When a vesicomyid clam is disturbed and withdraws its foot from the sediment, it is severing itself from the chemical source that sustains its bacteria, at least briefly. The foot goes out again as soon as the disturbance passes. The bridge reopens, the sulfide flows. In that reopening, there is no urgency felt by the animal, no anxiety of interruption. The clam extends its foot because that is what its body does, as the blood circulates because that is what the heart does, as the bacteria in the bacteriocyte cells of the gill process sulfide, because that is what those bacteria do. The whole is a system of embedded processes, each running at its own pace, each nested inside the others, the chemistry at the bottom driving the biology at the top, and the biology at the top structuring the chemistry at the bottom, down through the sediment surface, where the foot meets the pore water, all the way to the sulfate methane horizon far below. The Pravana snail, grazing across the white bacterial mat in the cold stillness, is part of this too, though at a distance from the deepest chemical layer. Its thick shell presses lightly against the mat surface as it moves. The radula works. The mat, which is itself a living structure built of filaments that have been running continuously on the chemistry of the sediment below, gives up some of its mass to the snail, which converts it into snail tissue, which will eventually become food for something else. The mat regrows. The snail moves on. The seep does not notice either of them. The chemistry continues below, indifferent to the grazing at the surface, indifferent to the scavenging crabs at the margins, indifferent to the instruments of whatever submersible has descended to document it. There is a stillness to these animals that is different from the stillness of animals at rest. A sleeping animal holds tension in its body, a readiness. These animals are genuinely slow. The tube worm does not flee. The mussel does not chase. The clam does not pursue its sulfide. It extends a foot into the place the sulfide already is. Their lives are organized around staying in the right place, maintaining the right chemical access, housing the right bacteria at the right concentrations. Motion is not their primary mode. Patience, if the word can be used without implying a mind, is the pain bee worms in a large aggregation have been in place for a very long time. Some of the oldest living colonies at cold seeps have been estimated at more than 2.5 centuries in age. Those tubes that have been standing since before the industrial era, before the first telescopes were pointed at the sky from the continents above, rooted in the same sediment, sustaining the same bacteria, drawing the same sulfide through the same blood molecule across a lifespan that has no parallel in most of the ocean. The colony changes over that time. Individual worms die and are replaced. The carbonate rock builds up around the tube bases. The sediment reorganizes around the accumulated mineral, but the aggregation persists, old and slow and continuous. Around the oldest tubeworm aggregations, the carbonate rock is often thick and pale. It is formed from the chemical alteration of pore water by the same sulfate-methane reactions happening below, the alkalinity of those reactions precipitating calcium carbonate mineral into the sediment over decades, slowly hardening it into rock. The rock then provides hard substrate that other organisms can attach to. Sponges, cold water corals, serpulid polychaetes in their own small mineral tubes. The seep, by precipitating its own geology, creates structure for organisms that have nothing to do with the chemistry of methane and sulfide. It builds a small reef, in a sense, not through biological secretion as in the tropics above, but through chemical deposition from below. The bathymedoilus mussel bed also accumulates its own geology. Dead shells pile up. Shell material forms carbonate substrate. Over long years, the accumulated shell layer becomes a hard surface that overlies the original soft sediment of the seep site, and the living mussel bed now rests on the record of previous mussel beds beneath it. To look at the stratigraphy of a cold seep site is to look at time made physical, the chemical record below, the mineral record at the surface, and the biological record in the organisms living and dead across the top. A Galathiid crab picking through a pile of dead mussel shells at a cold seep is, without knowing it, walking across geological history. The shells beneath its feet were once other mussels, each one housing the same dual endosymbiosis, each one reading the same fluid chemistry, each one building its dark shell from calcium dissolved in the water and dying in that dark water and contributing its shell to the accumulation underfoot. The crab does not know this. It is looking for soft tissue, finding scraps, moving on. But the shells it crosses are a record. They always have been. Deep in the sediment below all of this, below the mussels, below the clams, below the carbonate rock, below the microbial mats, below the frenulate worms in their thread-like tubes at the seep margin, the same fluid is rising. The same methane, dissolved in pore water, moving upward through the sediment along the chemical gradient toward the seafloor. The same sulfide, produced by the reactions at the transition zone between the upward diffusing methane and the downward diffusing sulfate, flowing up through the sediment to emerge at the surface and meet the organisms built to use it. The whole animal community above is organized around this upward flow. The tubeworm's root extensions reach down toward it. The clam's foot pushes into it. The mussel bed sits at the sediment surface where it becomes available. The bacterial mat tiles the very opening from which it emerges. Every body at the seep is positioned, in one way or another, relative to this invisible upward movement of reduced compounds through the sediment. The fluid does not rise quickly. It moves through pore water at rates that vary widely across the seep, from very slow diffusion at the margins to advective flow at the active center. But even the most active seep center is a quiet thing by surface world standards. The bubbles that rise from the most active vents, small spheres of methane gas departing the sediment one by one, catching whatever instrument light falls on them, and rising through the water column above in slow strings, are the most visible expression of this flow. But they represent a small fraction of the total methane transport. Most of the methane that moves through the sediment moves as dissolved gas in pore water, unseen, and most of it is consumed before it reaches the water column at all. What reaches the seafloor surface is the portion that escapes microbial consumption below, and what the animals and bacteria of the surface seep community encounter is that portion, the chemical fraction that has made it through the sediment filter and arrived at the interface between rock and water. Even this arrives slowly. The seep is not a jet. It does not pulse or surge. It seeps. That word is exactly right. The organisms built for this environment are organisms built for the slow and the steady. A vestimentiferan tubeworm growing at a few centimeters per year, anchored to the same sediment, drawing the same compounds through the same blood molecule, housing the same bacteria in the same organ, is expressing a version of life shaped by the pace of its chemical source. The pace is geological. The animal approaches it with a patience, a metabolic steadiness that has no analog in the fast-moving sunlit world above. The bathomodiolus muscle, responding to shifts in local fluid chemistry by adjusting the relative activity of two bacterial populations in its gill, is also a kind of instrument, a living chemical sensor, slow and soft-bodied, calibrated by millions of years of selection to read the fluid coming off the seafloor. The vesicomoid clam, transmitting its bacteria from mother to offspring through the egg, has solved the problem of continuity, how to ensure that each new generation carries the same chemical tools its parents needed by making the bacteria part of the inheritance itself. The snail's thick shell is the seep's acid, written in calcium and protein. The galotzid crab's pale color is the dark's answer to the question of pigment. Each body at the seep is in this way a record of the seep. Each animal shape, the long white tube, the oval dark shell, the pale thick shell of the snail, the heavy pale shell of the clam, the thin thread-like worm in its transparent tube, is the accumulated physical record of what these animals have needed to be in order to live here. The chemistry of the seep is written in them as completely as it is written in the carbonate rock, and has been being written for just as long. A cold seep on the continental slope, seen from above in the footage of a remotely operated vehicle, descending through dark water, resolves slowly from a general darkness into a pale, irregular patch of color. White mats, dark shells, pale rock, red plumes, if the tubeworm colony has extended above its tube openings. Then, as the vehicle settles close to the surface, the detail comes in, the texture of the Beggiato mat, the individual muscles with their overlapping edges, a Provanus snail at the margin of the mat, a crab moving unhurriedly through a field of empty shells. And below all of it, invisible, the fluid still rising, the methane still moving, the sulfide still diffusing upward through the sediment, a few millimeters per year or a few centimeters, depending on where the flux is highest, making its way toward the animals above that have been waiting for it, if waiting is the right word for a process that has no awareness of waiting, since long before this submersible was built, long before the ocean was mapped, long before the first person stood at a continental shore and looked out at the water, wondering what was below, the red plumes of a tubeworm extend into the dark water above the sediment and drift very slightly in whatever slow current moves at that depth. They are the most visible living thing at the Seep site. They catch the light of instruments better than anything else there, and their color, that deep red, dense with hemoglobin carrying sulfide and oxygen in careful chemical balance, holds against the dark water behind them like something warm in a cold place. They are not warm. The water around them is near freezing, but the chemistry running inside them is active, continuous, and ancient, and if warmth can mean ongoing process, the slow and the persistent and the enduring, then perhaps the color is right for what it signals. The symbiosis the tube worm carries inside it has been passed forward through time in an unbroken line of animal bodies, each one sheltering the same bacteria, each one solving the same chemical challenge with the same inherited molecules. The clam's bacteria, transmitted through the egg from mother to offspring, have traveled the same way, forward through generations, never entering the environment, never needing to, contained always within the tissue of the animal that has carried them. That continuity, bacteria inside animal, animal carrying bacteria, generation after generation, in the cold and the dark of the deep ocean floor, is its own kind of endurance. Not the endurance of the rock, which is passive and mineral, but the endurance of the living thing, which is active and warm in the only sense that matters, still running, still carrying, still doing the work. The seep continues below the animals continue above the seep. The carbonate rock continues forming, mineral by mineral, at the margins of the active vents, preserving the chemical signature of the reactions below, in a record that will outlast any individual organism living at the site. The mussels above will die and add their shells to the layer of past mussels. The tube worm colony will eventually exhaust its chemical source as the fluid pathway shifts, and it will fade, and the pale encrusted tubes will remain on the sediment as the slow mineral record of a colony that lived for centuries. The clams will retract their feet for the last time, and the valves of their heavy shells will part, and something will come to eat what is left, and the shells will settle into the accumulation. And below all of this, still, the methane will rise. It will find new paths through the sediment as old ones close. It will emerge at a different point on the seafloor, a few meters away or a few kilometers, and the mats will form there, and the mussels will settle, and the clam larvae will descend from the water column and push their first small feet into the new sediment, and the worm larvae will settle and begin their tubes, and the whole arrangement will begin again. Not the same individuals, the same arrangement, the same chemistry, the same inherited solution passed forward through time in the bodies of animals that have never seen the light, living off the slow exhalation of a buried world that has been breathing upward through the seafloor for longer than any individual life can measure. In the cold water above a mussel bed, above the pale carbonate rock, above the mat and the clam field and the worm colony with its red plumes drifting, a single mussel shell lies open on the sediment, empty now, its dark valves parted and still. The bacteria that lived in its gill have dispersed. The carbon they fixed has passed into something else. The shell will remain, adding its small mass to the geology of the place, becoming substrate, becoming record, becoming part of the slow mineral accumulation that marks the seep's long presence on this margin of the deep ocean floor. The water above is very cold and very still. It has been this cold for a long time. Something will settle here. Something already has. The seep continues, below, quietly in the dark, from directly above, in the cold instrument light of a submersible hovering in still water. A cold seep looks like a painting made in rings. At the center, dense and pale, a field of white stretches across the dark sediment. Mats of bacteria so thick they look almost solid, and packed among them the dark shells of mussels pressed together in the thousands. Around that center, a second ring begins, where the white thins, and the shells grow fewer, and clam beds take over, and the sediment between them carries the slow trails of snails moving across the seafloor in no particular hurry. Further out still, the life grows sparser, and the trails disappear, and the dark plane of the deep ocean floor returns by degrees, until there is nothing left to mark the seep at all, just sediment, and cold, and the immense weight of the water above. This is what a cold seep looks like from the outside, a set of concentric rings, each one a different chemistry, each one a different community of life, all of them radiating outward from a place in the seafloor where something is rising from below. The rings are not fixed. They shift over years and decades as the pathways through which fluid moves underground change course, widen, narrow, or close. What was seep center becomes seep margin. What was seep margin becomes open seafloor. The community follows the chemistry, and the chemistry follows the geology, and the geology follows pressures and faults, and the slow movement of sediment under its own weight far below. The whole pattern breathes in a rhythm measured not in seconds, but in years and centuries and longer. At the center of the rings, where the fluid flux is highest and the chemistry most extreme, the life is densest and also the most recently established. Microbial mats spread first, when fresh fluid finds a new path to the surface. Then the mussels come, colonizing the active vent sites in tight aggregations, their shells dark and their gills dense with bacteria. The mussels are among the fastest to arrive and among the first to thin as conditions shift. They live on the active edge of the seep, where the chemistry is richest and also least stable. Over time, as mats spread and mussel beds establish, and the first carbonate rock begins to harden in the sediment, something else moves and that will outlast all of them. The tube worms come slowly. They settle as small larvae into sediment that already carries a chemical signature, sulfide rising, methane in the pore water, the invisible signal of a functioning seep, and they begin to grow. Growth, for a tube worm, is not fast. In the cold of the deep ocean, where temperatures sit within a degree or two of freezing, where pressure is immense and food in the conventional sense does not exist, the rate at which a tube worm adds to its length is close to what a human fingernail adds in a month. A single tube worm might take decades to reach a meter. A colony, rooted together in the same sediment, might take a century to reach what a researcher measuring from the surface would call full size. And then it keeps growing. It keeps growing for longer than almost any animal alive. Some tube worm colonies at cold seeps have been estimated, through careful measurement and chemical analysis of their tubes, to exceed 250 years in age, 2.5 centuries. The sediment around them has changed in that time. The fluid pathways below them have shifted. Mussels have come and gone in the seep center nearby, and the bacterial mats have moved, and the carbonate rock has hardened around the bases of their tubes and become part of the permanent structure of the seafloor. Through all of it, the tube worms have stayed. Their white chitinous tubes, pale and encrusted and thick with age, anchor them to a place they cannot leave. Their red plumes extend upward into cold water, just as they have done for longer than any living person can remember, gathering oxygen and sulfide, feeding the bacteria inside them, growing by fractions of an inch each year, with a patience that has no human equivalent. A colony that old has been part of the seep for longer than the United States has been a country. It has been there since before any instrument reached the deep ocean floor. It has been there, in the dark, in the cold, in the quiet, with no witness and no need of one. Around it, the seep community has cycled through phases: the early rush of mat and mussel, the gradual hardening of carbonate, the slow establishment of the tubeworm aggregation itself, and the colony has occupied each phase in turn, without awareness of any of them, simply growing, simply filtering, simply converting sulfide and oxygen into more of itself. This cycling of phases is what ecologists call succession. On land, succession is visible. A cleared field becomes a meadow becomes scrub, scrub becomes forest. The progression takes decades at most, and can be watched across a human lifetime. At a cold seep, the same logic applies, but the timescale stretches past what observation can easily hold. The early stages, mat and mussel, last years to decades. The tubeworm stage lasts centuries. And beyond the tubeworm stage, when the fluid flux has dropped and the active vent has quieted and the mussels and their bacteria have found other sites, what remains is the carbonate. Pale, irregular, precipitated mineral by mineral from fluid altered by the chemistry of the sediment. The carbonate rock hardens the seafloor around and between and beneath the tubes that anchored there for so long. When the tubeworms are finally gone, their absence is written in stone. The carbonate rock that cold seeps leave behind is not just a record of one seep, in one phase, in one century. It is a record that can be read by those who know how to read it across geological time. The chemical signature of anaerobic methane oxidation, the specific ratio of carbon isotopes, the alkalinity that drove mineral precipitation, the sulfide minerals embedded in the matrix, is written into the rock at the moment of its formation, and it does not fade. Carbonate rock precipitated at a cold seep on the seafloor of the deep Pacific today carries the same chemical fingerprints as carbonate rock formed at a cold seep in the deep ocean of the Cretaceous period, when the continents were in different positions and the seas were warmer and the creatures living in them were almost entirely different from any alive now. The rock does not care. It records what happened, and it holds it. Ancient seep carbonates have been found in rock formations, now exposed at the surface in California, in Japan, in Scandinavia, seafloor deposits uplifted over millions of years by the movement of tectonic plates. Now sitting in cliff faces and road cuts and coastal outcrops where someone with the right training can press a hand to the stone and read in its chemistry the record of a cold seep that was active 100 million years ago. The mats were there, the mussels were there, or their ancestors, with the same bacteria in their gills, the same dual symbiosis, the same dependence on sulfide and methane. The tubeworms were there, rooted in their sediment, feeding their internal bacteria, growing slowly through the cold dark. None of them are there anymore. The rock is there, pale and layered and mineral dense, holding the record of their chemistry without any awareness of what it holds. 100 million years is a duration that does not fit inside ordinary comprehension. It is easier to approach it from the side. When those seeps were active, when the mats were white and the mussels were dense, and the tubeworms were extending their plumes into water that was cold, in the same way the deep ocean is cold today, the creatures walking on land above included dinosaurs. The Rocky Mountains had not yet fully formed, no flower had yet evolved. The planet was recognizable as Earth in its broad outlines, ocean, atmosphere, continental crust, but its surface was another world. And somewhere below that surface, in the deep ocean that no eye had ever seen, cold seeps were operating exactly as they operate now. The mats were spreading, the mussels were filtering, the tubeworms were growing their slow increments, the carbonate was hardening, and when those particular seeps quieted and the fluid paths closed and the organisms died, the carbonate remained, carrying the record forward in time through 100 million years of burial and compression and uplift until it reached a surface where a geologist could find it and read it. This is the continuity that cold seeps represent. Not the continuity of individual organisms. No mussel or tube worm alive today has any direct link to those Cretaceous seeps, but the continuity of an ecological form, a type of community, a way of being alive in a place where sunlight has never reached and where the chemical energy that drives all life rises from below. The form has persisted. The conditions that create it, continental margins, buried organic matter, upward migrating methane, the cold and pressure of the deep ocean floor, have been present on Earth for as long as there have been oceans and continents and organic matter buried in seafloor sediment, which is to say, for most of the history of complex life. But the seep community is not sealed inside itself. It is connected by chemical signals in the water, by the movement of animals, by the physical transport of tissue and organic matter to the broader deep ocean that surrounds it. Mobile animals come to the seep to feed. Rat-tail fish, long-bodied and pale, move through the outer margins of seep communities with a slow, unhurried grace, pausing near mussel beds and carbonate outcrops, feeding on the animals gathered there. Cusk eels thread through the structures of the seep in a similar way. Small zorisid fish, the eel pouts, occupy the quieter margins, feeding on worms and crustaceans and whatever else the seep's productivity supports. These fish are not chemosymbiotic. They do not carry sulfide oxidizing bacteria in their tissue or methane oxidizing bacteria in their gills. They are ordinary predators and scavengers from the general deep sea ecosystem, drawn to the seep because the seep is a place of unusual density and energy in an ocean floor that is otherwise very sparse. What they carry away matters. The carbon in a mussel's gill tissue, the carbon that was fixed there for methane or sulfide by a bacterial endosymbiont, becomes, when a rat-tail fish eats that mussel, carbon in the fish's muscle, and that fish moves away from the seep back into the open deep sea floor, carrying in its body atoms that were methane in the sediment days or weeks before. It carries the seep's productivity with it, distributing it outward into the wider ocean in a slow, steady, invisible transfer. Stable isotope studies, analyses of the specific ratios of carbon and sulfur atoms in the tissues of consumers have confirmed this. The chemical signature of chemosynthetically fixed carbon, with its distinctive isotopic fingerprint, appears in the bodies of fish and crustaceans and other mobile animals collected at the edges of seep communities and at some distance from them. The seep feeds the fish feeds the deep sea floor. The transfer flows in both directions. Animals from outside the seep bring organic matter into it, their waste, their shed tissues, eventually their bodies. The seep community's food web is not a closed circle. It is a node in a larger network connected to the sediment around it, to the water column above it, to the organisms drifting through that water column and sinking to the floor when they die. A seep community processes the chemistry rising from below and exports some of that energy outward into the ocean. It also receives input from above, from the slow rain of particles that falls through the water column from the sunlit surface far above. Though that input is a small fraction of what the seep generates internally. The seep is, in energetic terms, largely self-sufficient, but it is not isolated. The connectivity between seeps and other reducing habitats in the deep ocean is one of the stranger aspects of these communities. A cold seep on the Gulf of Mexico slope is separated by thousands of miles of open deep sea floor from a cold seep on the Pacific margin of Japan. Between them lies an ocean, much of it cold, dark, and nearly empty of the chemical signals that the specialized fauna of seeps need to survive. And yet the organisms living at those two widely separated seeps belong to the same families, and in some cases, the same genera: Vosicamaid clams, Bathymodilus mussels, vestimentiferan tubeworms, the same symbioses, the same body plans, the same chemical dependencies. The question of how they got from one place to another, how the larvae of animals that depend on very specific chemical conditions manage to cross thousands of miles of open ocean where those conditions do not exist, occupied researchers for decades. The best current picture involves stepping stones, hydrothermal vents, or similar chemosynthetic communities form around very different thermal and chemical conditions, represent one type of stepping stone. Whale falls, the bodies of large whales that sink to the deep sea floor and become, in their long decay, islands of sulfide production created by the anaerobic decomposition of lipid-rich bone, represent another. A whale skeleton on the deep sea floor, in the later stages of its decay, supports communities of sulfur-oxidizing bacteria, small polycate worms, and in some cases the same specialist fauna found at cold seeps and hydrothermal vents. Some subogolinid worms, some vesicomiid clams, some Bathymodilus mussels, not present at every whale fall, but present at some, and present in quantities sufficient to produce larvae that could potentially cross the open floor to a new reducing habitat. The whale fall is a bridge, temporary, unpredictable, but real. It appears when a whale dies and is consumed, and it lasts for decades, and during those decades it offers a foothold where the next generation of deep-sea chemosymbiotic fauna can settle and grow and release their own larvae into the current. Between whale falls and cold seeps and hydrothermal vents, the deep ocean floor is speckled with reducing habitats at a density that across geological time would have provided enough stepping stones for populations to spread around the world. Not quickly, not directly, but over the timescales that evolution works with, thousands and millions of years. The distance between a seep off the coast of California and a seep in the deep Pacific is crossable, one larval generation at a time, one ephemeral sulfide island at a time. Molecular phylogenetic studies support this picture. The evolutionary relationships among subogolinid tubeworms, vesicomiid clams, and Bathymodilus mussels, traced through the branching patterns in their genomes, show histories that connect populations separated by ocean basins, populations that, if you looked only at the present-day map, would seem impossibly far apart, but that share ancestors who made the crossing over time. This is the network in which the cold seep exists, not a solitary feature on a featureless floor, but a node in a system of nodes, connected by the slow movement of larvae and the slow succession of temporary habitats across a deep ocean that has been rearranging its floors and its margins and its chemistry for hundreds of millions of years. The seep is ancient, the network is ancient. The form of life it supports, chemosynthetic, symbiotic, sustained by what rises from below, is among the oldest ecological forms in the deep ocean, and possibly among the oldest on the planet. It persists because the conditions that create it persist. As long as there are continental margins, there will be sediment accumulating on them. As long as there is sediment accumulating, there will be organic matter buried within it. As long as there is organic matter buried in oxygen-depleted sediment, microbes will convert it to methane. As long as there is methane in the sediment, and pathways through which it can migrate, it will move upward toward the sea floor. And at the sea floor, where the methane meets the ocean, the seep community will form. The mats will spread, the mussels will colonize, the tube worms will settle and begin their long, slow growth, and the carbonate will start precipitating, mineral by mineral, from pore water altered by the chemistry of the sediment below. The whole cycle will begin again, as it has begun again at every active seep site, on every continental margin, on every ocean floor, since long before there were humans to find it. And the seep will operate in the dark. It will operate in the cold. It will operate under pressure so immense that no unprotected living thing from the surface could survive it, and in a silence so complete that no sound from the surface ever reaches it, and at a depth where the light of the sun, 8 minutes away from the surface, but absent here entirely, has never, in the entire history of this particular patch of seafloor, penetrated even a fraction of the water column above it. None of this is an obstacle for the life that lives there. The darkness is not experienced as darkness. The cold is not experienced as cold. The pressure is simply the condition of existence, as unremarkable to a vesicomiid clam as the pressure of the atmosphere is to the birds that move through it. The seep community does not find its environment extreme, it finds its environment home. There is something in that which settles the mind. The long history of these communities, the hundreds of millions of years during which the same ecological form has been expressing itself on the same continental margins, under the same cold pressure, in the same complete absence of sunlight, is not a history of struggle or endurance. It is a history of fit. The organisms that live at cold seeps are not surviving despite their environment. They evolved within it, and for it, over timescales so long that the fit between organism and environment has become seamless. The clam's foot reaches into the sediment because the sediment holds sulfide, and the sulfide feeds the bacteria in the clam's gill, and the bacteria fix carbon that becomes the clam. The mussel filters water across gills, seeded with 2 different microbial populations, because the water above an active seep carries both methane and sulfide in varying proportions, and the dual symbiosis allows the mussel to use whatever is more abundant. The tube worm has no mouth because the bacteria in its trophosome feed it everything it needs, and so the mouth became unnecessary, and over millions of generations it was simply not there anymore. These are not compromises, they are refinements, shaped by time into something exact. The carbonate rock that forms around these communities and preserves their chemical record is also, in a quiet way, a form of permanence. A seep that has been active for centuries will harden its surroundings as it operates, the rock precipitating slowly around the bases of the tubeworm tubes, filling the spaces between mussel shells, creating a substrate of pale, irregular stone that persists long after the fluid flow has stopped and the organisms have gone. The hardened carbonate changes the habitat. It creates surfaces for encrusting organisms, for filter feeders that prefer hard substrate to soft sediment, for corals and sponges and the other animals of the deep that need something solid to attach to. A seep that has quieted is not empty. It has become a different kind of habitat, dense with organisms that take advantage of the structure the seep left behind. And within the rock itself, locked into the mineral fabric, is the isotopic record of what the seep was, the methane it processed, the sulfide it generated, the specific chemistry of its microbial communities. The rock holds the memory. It carries it forward into whatever geological future awaits this particular piece of seafloor. In the rock formations now exposed on land, in the cliffs of California, in the outcrops of Japan, in the pale stone of Scandinavian coastlines, Those memories are now readable. Researchers press instruments to the surface of ancient seep carbonates and measure the ratios of carbon isotopes in the mineral, and from those ratios, they read the chemistry of a seafloor that existed before humans walked the planet. The seep is gone. The organisms are gone. The continental margin that hosted it has been deformed by tectonics and uplift and erosion over 100 million years into something unrecognizable. And still the chemistry is there, in the rock, clear enough to measure. There is no record of what it felt like to be there. No record that feeling was even present. But the chemistry is exact, and from it, the form of the community can be reconstructed. The mats that were white, the muscles that were dense, the tube worms that were growing their slow increments in water that was cold and dark and full of sulfide. These ancient seep communities were processing methane. They were filtering it from the sediment below and consuming it at the sea floor and converting it through layers of microbial chemistry into the bodies of animals and the shells of clams and the fabric of carbonate rock. The same process is happening right now on the continental margins of every ocean on the planet. The Gulf of Mexico slope, a few hundred miles from the nearest city, hosts seep communities that have been active since before the first European arrived in the Americas. The deep margins of the Pacific, thousands of miles from any shoreline, host seeps that have been operating for longer than the current configuration of the continents has existed. The Hadal Trenches, where the seafloor drops to depths that exceed 5 miles, host seep communities in the most remote and pressurized environments on Earth, in places that human-operated vehicles have visited only a handful of times. The seeps are everywhere. The conditions are right. The conditions are right, in one form or another, on every continental margin on the planet. There is a quality to this that is not easy to name. It is something like steadiness. It is the quality of a process that has been running so long in so many places, under so many variations of local geology and chemistry and depth and temperature, that it has become part of the basic texture of the planet. The deep ocean floor is not empty. It is not inert. It is not simply a dark, cold, featureless expanse of sediment where nothing happens. It is alive in places with communities that have been there longer than most of the surface features of the Earth. The mountains that border the coasts above these seeps are younger than the seep communities below them. The rivers that carry sediment to the continental margins and feed the buried organic matter that will eventually become methane are younger than the ecological form of the communities that depend on it. And in the rock, in the sediment, in the slow movement of fluid through fault and fracture and porous layer toward the seafloor, in all of this the process continues, as it has always continued, as it will continue, long after the current configuration of life on the surface has changed beyond recognition. The methane rises, the mats spread, the mussels colonize, the tube worms settle and grow and extend their red plumes into water that is cold and dark and still. The carbonate hardens slowly around them, and in the deep, quiet dark of the ocean floor, at the center of the concentric rings that a submersible's light would reveal if it were there to look, the seep breathes in its long, slow way, not aware of its own antiquity, not aware of the rock record it is adding to, not aware of the fish moving through its outer margins, carrying its carbon into the wider world. The seep simply operates. It operates in the way that it has always operated, and the way it will continue to operate in the deep, cold dark, when the light of any instrument has long since turned and risen and gone.